Good day, class. I hope you're good. I'm fantastic myself. I'm very excited for today's lessons because we're doing my favorite part of tutoring, which is answering questions. Right. I enjoy answering questions because application is more important than theory. I'm not saying that theory is not important, but the application of knowledge is more important, right? So let's start. Before wasting any time, just get into it, right? So today we'll be doing the November 2024 question paper, right? Is it a Gauteng? I think it's a Gauteng one. Just pick the random question paper, right? Just pick the random question paper so that we can do it before you even write your final exam. For those who are writing their final exam this June, I'm speaking about those who are upgrading, right? So we'll just go straight to the questions without wasting any time. Okay, now let's get to question one. So question one, I won't read the statement there. I'll just go into the question. You already know the statement. It's a multiple choice. You know how you're supposed to answer, right? I'm just going to get into the question and help you assist you on how to approach such questions. And bear in mind, question one is very important. Multiple choice is very important. You have 10 questions, right, with 2 marks each, with a total of 20 marks, right? So you cannot miss that 20 marks. You just have to make sure that you get the 20 marks, right? Okay, so 1.1 reads as follows. The hormone responsible for the regulation of salt content in the human body is A, testosterone. We know it's not testosterone, right? Testosterone is not responsible for salt content in the body, right? Let's go to B, aldosterone. Yes, aldosterone is responsible for salt, regulating salt content in the body, right? As for testosterone, is not responsible for that. Remember, what are the functions of testosterone? Functions of testosterone, they trigger meiosis. They're responsible for meiosis, and also they're responsible for secondary characteristics in males. Does that make sense? Secondary characteristics in males, or puberty, or you can say puberty. Let me just say puberty and simplify it, right? C cannot be C. C is prolactin. Remember, prolactin is responsible for milk production, right? It's responsible for milk production in females. Does that make sense? D, glucagon. Glucagon is responsible for sugar regulation in the body, or glucose regulation in the body. It's not responsible for salt content. So our answer here is B. Let's go to 1.1.2. Which one of the following is a stage in human embryonic development? A, choriod. We know that's not the answer, right? It's not even a stage. What is a choriod? A choriod is a layer, it's the second layer of the eye. So it deals with the eye. So really, it's out of the question, right? B, amnion. What is an amnion, right? An amnion is a membrane, right? It's a membrane in the amniotic egg, right? So it's the first membrane in the amniotic egg that encloses the amniotic fluid. Whether we're dealing with reproduction in vertebrates or we're dealing with reproduction in humans. Does that make sense? So amnion is out of the picture because that's a membrane. C, morula. Yes, morula makes a lot of sense, right? Remember, we're starting with our zygote, right? Starting with fertilization. Let me explain the whole process. So we're starting with fertilization. After fertilization, we have a zygote. After the zygote divides by mitosis. After dividing by mitosis, it forms what? The morula, right, which is a ball of cells. The morula further divides by mitosis to form a hollow ball of cells. What is that hollow ball of cells? The blastocyst, right? And the blastocyst will go to the endometrium to attach there or to implant. Does that make sense? So morula is definitely a stage in the human embryonic development. So the answer is C. It's definitely not D, because D is the chorion. What is the chorion? Chorion is also a membrane, right? It's also a membrane. Let's move on. 1.1.3. When the carbon dioxide level in the blood increases above normal, A, the rate and depth of breathing decrease. We know that's not true because when the levels of carbon dioxide increase in the body above normal, the rate and the depth of breathing increases instead of decreased, right? So that more oxygen is drawn to the lungs, right? For purposes of exhaling it out. Does that make sense? Okay, A is not the answer. Let's go to B. So B reads as follows. Receptor cells in the diaphragm are stimulated. Receptor cells in the diaphragm. We know that's not true. Receptor cells are not in the diaphragm. Where are receptor cells that are responsible for detecting carbon dioxide levels? They're in the carotid artery in your neck, right? So chemoreceptors, we call them chemoreceptors, and they are in your carotid artery. Does that make sense? Okay, so B is not the answer. Let's go to C. Heart beats slower. We know that's also not true because the heart actually does the opposite. It beats faster whenever the carbon dioxide levels they increase in the body. It beats faster, right? So C is not the answer, which leaves us with D. We don't even have to read D. Right? The answer is definitely D. Receptor cells in the carotid artery in the neck are stimulated. So D is our answer, right? Okay. Now let's go to 1.1.4. 1.1.4 reads as follows. Which one of the following changes occurs under the influence of adrenaline? Right? Adrenaline. So which changes occur in the body? So let's go to A. An increased blood supply to the skeletal muscles. Yes, because we call the adrenaline hormone as a flight or flight, right? Hormone. So whenever you're flying or whenever you're fighting, right, you need your muscles to have more oxygen, to have more energy, to have more blood supply because you're using them whenever you're fighting or whenever you're running, right? So A is definitely the answer. But let's look at other options. B, decreased blood pressure. Cannot be decreased blood pressure because blood needs to get to different parts of your body quickly because you need that oxygen. Remember, the blood also carries oxygen. So you need oxygen to get to those important critical organs and parts of your body so that you can run, rather, not right, so that you can run or you can fight. Does that make sense? Okay, let's go to C. Decreased muscle tone of skeletal muscles. No, which is the contraction. Muscle tone is the contraction and relaxation of your muscle. Definitely is not the answer. Actually, you need your muscle tone to be increased in instances or in moments where you are flying or fighting, right? It's weird how I'm saying flying, but you understand, get the drift, right? Okay, let's go to D. D, an increased blood supply to the digestive system. An increased blood supply to the digestive system. Note, actually, it's an increased blood supply to the digestive system so that most of the blood goes to your brain and other parts that needs it the most. So it does not go to the digestive system, right? Instead, it goes to the brain, it goes to the skeletal muscle, right? So it goes to the places where it's needed the most. So definitely D is not the answer. So let's go to 1.1.5. So 1.1.5 reads as follows. A girl is barefoot and steps onto a thorn, causing a reflex action. Remember, what is a reflex action? A reflex action is an automatic, rapid response to a stimulus, right? I'll repeat that again. It's an automatic, rapid response to a stimulus. What is a stimulus? Stimulus is a change in the external environment, right? Okay, now let's go to the question. Which one of the following represents the correct pathway of the reflex arc? So which one of the following? Let's look at A. Let's explore A. So A says that we start with the pain receptor, okay? Then the pain receptor, from the pain receptor we go to the motor neuron, which is not true. Already this answer is out of the picture. We're supposed to have a sensory neuron, right? Before the motor neuron. So it's wrong, right? Okay. Let's go to B. So B, foot muscle, it cannot be a foot muscle, right? Cannot be, because at that point you're not reacting, right? Because you use muscles in order to react. So it cannot be a foot muscle. The moment it starts with the muscle, it's wrong. It's supposed to start with the receptor, because the receptor is the one that receives the stimulus and then converts it to an impulse. Okay, let's go to C. C is already wrong because it's starting with the foot muscle. We're not supposed to start with the foot muscle. So it leaves us with D. So D is the correct answer, but let's explore it further. So D states that we're starting with the pain receptor, correct? The next one is the sensory neuron, correct? The next neuron is the motor neuron, correct? Then the last part, right, of the sequence is the leg muscle, which is our effector. D is the correct answer, right? So D is the correct answer. Okay, let's go to 1.1.6. 1.1.6. The diagram below represents the human eye. So whenever you encounter such questions, the important thing is for you to analyze the structure and the parts of the eye, right? Let's look at X. What is X, right? So our X becomes our ciliary muscle, right? Ciliary muscle, right? And then our Y becomes our suspensory ligaments. Our Y becomes our suspensory ligaments. And then finally our Z becomes our lens, right? Lens. Then after you've identified the parts, you state their functions. So let's look at the function of X. So the function of X, the X which is ciliary muscles. Don't confuse it with circular muscles. Please don't make that mistake. So X is ciliary muscles. What's the function of ciliary muscles? So the function of ciliary muscle is that they hold suspensory ligaments in place. So that's the function of ciliary muscles. Now let's go to the function of suspensory ligaments. What's the function of a suspensory ligament? So the function is that they hold, as you can see, that they are holding the lens in place. And also they play a role in altering the shape of the lens, right? Okay, now let's go to our lens. What's the function of our lens? As you can see, this biconvex structure. So the function of our lens is that it plays a role in the refraction of light. When I say refraction, I speak about bending, the bending of light, right? Or you can say that it allows light to pass through since it's a transparent structure. Okay, let's go to the question here. So we're done analyzing our structure. Let's go to the question. Do this. Okay. Okay. So as a spectator in a soccer stadium is seated 200 meters away from the field. Already we can analyze the question, right? That this is an object that's further away. Already you're thinking about accommodation. And already you're thinking about accommodation. What happens in the eye when someone is looking at an object that's further away? What happens to the ciliary muscles? So the ciliary muscles, they relax, they don't contract. The ciliary muscles, they relax, right? Then what happens to the suspensory ligament? The suspensory ligaments, they become tight or taut, right? And then what happens to the lens? Then the lens becomes less convex. So those are changes that happen when someone is looking at a picture that's further away. So let's go to options, right? Let's go to options. Or let's read the question first. Which one of the following describes the condition of structures X, Y, and Z when he looks at the ball placed in the middle of the field? Sorry about that. So let's look at A. A, relaxed. So muscle X is relaxed, correct, which is correct. Structure Y. So what is structure Y? Structure Y is your suspensory ligaments or are your suspensory ligaments. they become slack. We know that's wrong, right? Because you cannot have a relaxed ciliary muscles and then have slack suspensory ligaments at the same time. You can't, right? You can't, so it's wrong. Let's go to B. Muscle in X is contracted. Already it's wrong. It's out of the picture because muscle or ciliary muscles are relaxed instead of contracted. Now let's go to C. So C states that muscle X is relaxed, which is correct. Structure Y is taut or tight, which is correct. Then part Z is less convex. So the correct answer is C. Already we're ruling out D because it's starting with contracted. So the answer is what? C. If you don't remember this, go back and revise your accommodation processes, right? Okay, let's go to 1.1.7. So 1.1.7 reads as follows. Which one of the following is involved in thermoregulation? Okay, thermoregulation, remember, we are regulating temperature. The regulation of what? Temperature. Let's look at our options. A, corpus callosum. Wrong. Corpus callosum is the bone that separates the brain, right? Or the two hemispheres of the brain. The left hemisphere and the right hemisphere. So it's not correct, right? Let's go to B, hypothalamus. B, yes, we know that B is correct because the hypothalamus is the control center for temperature. Does that make sense? So the answer becomes what? B. So C, cerebellum is not correct. D, spinal cord is not correct. Let's continue. So let's go to 1.1.8, which reads as follows. The following is the list of factors regarding a developing fetus, right? Okay, let's look at Roman figure 1, temperature regulation, right? Number 2, protection. Roman figure 2, protection. Roman figure 3, free movement. Roman figure 4, nutrition, right? So let's go to the question. For which combination of factors is the amniotic fluid responsible? Obviously, as we look in these options, Roman figure 1 is correct, Roman figure 2 is correct, Roman figure 3 is correct, but Roman figure 4 is not correct, right? So the amniotic fluid is not responsible for nutrition. We have the placenta for that, right? So we have the placenta for that. So the answer becomes what? A. So our answer is A. Roman figure 1, Roman figure 2, and Roman figure 3. Okay. Now let's go to 1.1.9. So let's analyze our structure here, right? So this is the structure of the eye. Actually, this is our middle ear, right? We have our middle ear. They didn't show us the outer ear, which is formed by the pinna, auditory canal, and the tympanic membrane. They only showed us the tympanic membrane, which divides the outer ear and the middle ear. So this is the structure of the middle ear. Does that make sense? I hope it does. So let's analyze. So we have our A here. What is our A? Our A is our eardrum or tympanic membrane, right? Then we have our ossicles, the three ossicles here, right? We have our hammer, right? We have our anvil, and we have our stirrup, right? Stirrup, or you can say malleus, incus, or stipes. Okay, and let's focus on B. What is B? What structure is B? So B becomes your round window, right? It's just beneath the oval window. Then C becomes our Eustachian tube. C becomes our Eustachian tube. Let me just remove this. Right, C becomes our Eustachian tube. Okay, so let's look at the functions of each of these structures. What am I doing? Okay, let's look at the function of each of these structures. So let's start with our A. So our tympanic membrane. What's the function of our tympanic membrane? So the function of the tympanic membrane is that it transfers vibrations, right, to the middle ear or to the ossicle, right? We can say that it converts sound waves to vibrations and then transfers those vibrations to the ossicle, right? Then let's look at our function, the function of B, the round window. So what's the function of the round window? The round window absorbs excess pressure in the inner ear. So it absorbs excess pressure in the inner ear. Okay, let's go to our C. So C is our eustachian tube, right? What's the function of the eustachian tube? So the eustachian tube is responsible for maintaining or equalizing pressure on either side of the tympanic membrane. So we're done with the functions. Let's go to the questions. Right. The nicest thing is that you can replay this video over and over again if you did not hear me. Right. So try to move with me. Try to move with the same pace as me. Let's go to the question. Which one of the following occurs when there are excess waves in the inner ear? Okay. Excess waves. The moment it says excess waves, we know that we're dealing with the round window, which is structure B. Right. So anything that has structure B is the correct answer. So let's look at A. Reads as follows. Pressure is released at A. Pressure is released at A. We know that's not true. A is the tympanic membrane. Okay, let's go to B. Pressure is released at C. Yes, pressure is released at C, but we're speaking about excess pressure waves, right? We're speaking about excess pressure waves in the inner ear, right? Excess pressure waves. And C deals with the exiting, right, and the entering of A in and out, right, of the middle ear, right? So we're not speaking about that. Let's go to C. Pressure is absorbed at B. Yes, so the answer becomes what? B. So pressure is absorbed at what? B. Because we're speaking about excess pressure waves in the inner ear. And which part is responsible for absorbing those pressure waves is the structure B, which is your round window, right? Why is it absorbing excess pressure? In order to prevent echoing. Hope that makes sense. So D already is out of the picture because we already have our answer. So our answer becomes C. Okay, let's go to 1.1.10. So 1.1.10, the diagram below represents the transmission of impulses from a structure in the inner ear to the brain. Right? So we can see here already by analyzing the structure, I can see two processes. I can see the hearing part and I can see the balance part. So the balance part, right? Let's look at the part here. Obviously, they're showing us the part. So from E, so let me just draw this so that it's easier for you. So from E, right? Look at E. So it's pointing at the base of the semicircular canals, and we know at this point what we get here. We have our receptor called what? Cristae. This thing is just awful, man. It's awful, right? Writing with this thing is just not user friendly. Okay, so at the base of the semicircular canals, we have what? We have our cristae, right? Cristae, they deal with the change in the speed and direction, right? When the speed and direction of the head changes, the cristae are responsible for converting that balance stimulus to an impulse, right? Okay, and that impulse is taken, right, from E to A to H. That balance impulse is taken to H, which is our cerebellum. Does that make sense? So that's the balance one, right? I hope it makes sense, right? Okay. Now let's move on to our utriculus and sulculus. So this other arrow here, right? This other line segment is pointing. Let me just say line segment because it's a line segment. This line segment is pointing at our utriculus and sulculus. So here in our utriculus and sulculus, you find what? You find your maculae. Maculae deals with the change in the position of the head. When the position of the head changes, the maculae are stimulated, right? And the impulse moves in the same direction. It's E, right? E to H, right? You can also include F, right? So if F represents the auditory nerve, then the path becomes from E, F, and H, right? So that's balance. Okay. Now let's remove this. Okay. And also you have the process of hearing. So the process of hearing starts here. So it becomes your for your organ of Corti inside the cochlea, which converts the stimulus, the sound stimulus, to impulses. Then from J, we move to F, right? To F. Then from F, we move to G. So G is our cerebrum. So that's where hearing is interpreted, or where the impulse of hearing is interpreted, right? Okay. Okay. Let's erase this. Just erase it. Okay, so we're done with analyzing our picture, but let me leave it as is because it's going to waste time. Okay. So let's read the question. The question reads as follows. The nerve impulses for balance are transmitted as follows. For balance, already we know that for balance it's E, F, H. Let's look for EFH. So EFH is B, right? So B, E, F, H, right? EFH. So that's our answer. So our answer is B. Okay, now let's go to our biological terms, right? This is the most interesting part of the question paper for me, right? This is the most interesting part of the question paper. Let's go to 1.2. So 1.2 reads as follows. 1.2.1. I won't read the statement there. I'll just go straight to the question. 1.2.1. The structure that connects the fetus to the placenta. Obviously, that is, we know that is the umbilical cord. Umbilical cord, right? Not the umbilical cord. Umbilical cord. Umbilical cord, right? I don't know how you pronounce it, but I pronounce it as the umbilical cord. So remember, it's the cord that connects the fetus to the placenta. And remember, we said that when I was teaching reproduction uh, in my class, I was saying that the umbilical cord contains two, right? Two artery, two umbilical artery, and one umbilical vein, right? And the umbilical arteries, they deal with waste from the fetus, and the umbilical vein deals with nutrition from the mother, from the placenta to the fetus, right? Okay, so the answer here is the umbilical cord. Let's go to 1.2.2. The part of the nervous system that consists of sympathetic and parasympathetic sections, right? The part of the nervous system that consists of sympathetic and parasympathetic section, rather. So we know that the part of the nervous system is the autonomic nervous system, right? But I'll just try to categorize it for you. So we're starting with our nervous system. Our nervous system is divided into two, divided into the central nervous system and the peripheral nervous system, right? Then our central nervous system is divided into two parts, divided into the brain and the spinal cord. So we're done with the central nervous system. Now let's move to our peripheral nervous system, or the PNS, right? So peripheral nervous system is divided into two. It's divided into the somatic nervous system and the autonomic nervous system. The autonomic nervous system is then divided, right, subdivided into two parts, divided into the parasympathetic nervous system and the sympathetic nervous system. So our answer here is the autonomic nervous system, right? Okay, now let's go to 1.2.3. So 1.2.3 reads as follows. Receptors in the ear that detects changes in speed and direction of movement of the head, right? Whenever they're speaking about speed and direction, right, they're speaking about the cristae or crista. You can say crista or cristae. Right, so the answer here is cristae. Let's go to 1.2.4. The ossicle that transfers vibration to the oval window. Okay, already we know the answer that the only ossicle that's connected to the oval window is the stepes or the stirrup. Right, we can either say the stepes or the stirrup. That's the answer there. Let's move on to the next question. 1.2.5. So 1.2.5 states that the growth reaction of a plant in response to gravity. So we know that the growth response of a plant in response to gravity is geotropism. Geotropism. Does that make sense? Or we have the other one, right? We have the other response, right, which is not mentioned here. The other response is responding to light, which is phototropism. So go back to your plant response and read through that, right? If you've already forgotten, rather, right? So go back to that. So the answer here is geotropism. Okay, let's go to 1.2.6. The part in the inner ear where the organ of Corti is located. Again, this is simple. Where the organ of Corti is located within the cochlea, right? The cochlea, that snail structure, snail-like structure, right? Because it's shaped like a snail, like the back of the snail. Okay, let's go to 1.2.7. A plant hormone that stimulates the germination of seeds. So what is germination? Germination is when the seeds they start to sprout, right? So what's responsible for that? You can say the gibberellins, right? Gibberellins are responsible for the germination of seeds. So gibberellins. Okay. Remember, we have three plant hormones. We have the auxins, abscisic acid, and gibberellins, right? Abscisic acid actually is the opposite of gibberellins. Where one promotes seed germination, or where one stimulates seed germination, the other inhibits seed germination, right? In the case of abscisic acid inhibits or prevents seed germination during winter, right? Or during times where water is scarce, right? Okay, so the answer here is gibberellins. Let's go to 1.2.8. The structure in the head of the sperm cell that contains enzymes, right? Obviously, we know that that structure is called acrosome. Acrosome. So what's the function of that acrosome? It's responsible for penetrating the walls of the ovum. Let's go to 1.3. So 1.3. So we already know the rules in 1.3. Please make sure that you don't write A, B, A and B. Please follow the instructions. You either write A only, B only, both A and B, or none. So those are the rules. Right? Other than that, you get it wrong. Okay, let's go to 1.3.1. So 1.3.1. Let's read the question. Acts as a microfilter for the developing fetus. Okay, microfilter obviously is A only, it's the placenta. Right? It acts as a microfilter because it allows waste, right, and sends out nutrition. So it acts as a microfilter in that manner. It's definitely not the cervix. Definitely not B, right? It's definitely not B. I hope that makes a lot of sense, right? Okay, we just had a shutdown of electricity here. I just had a shutdown of electricity, but let's continue. Let's continue, right? Let's continue. Okay, 1.3.2. So 1.3.2. So the part of the brain that controls the heart rate. The part of the brain that controls the heart rate. So what's the answer there? So 1.3.2. So the answer there becomes B only. Your B only. This is the medulla oblongata. Right? Medulla oblongata. Let's go to 1.3.3. 1.3.3. The area on the retina where there are no rods or cones. So the answer there is A only, the blind spot. Right? Remember, in the blind spot, we don't have rods, no cones. Okay. Okay, 1.4. 1.4. Let's go to the questions 1.4. Right, 1.4.1. Give the letter and name of the structure that forms part of both the urinary and the reproductive system. So what's the answer? So the answer there becomes C. So what is our C? Our C is the urethra, right? Because remember the urethra, what's the function of the urethra? Function of the urethra, it's a passageway for both urine and sperms or semen. Right? Let me just say semen. Okay, let's go to B. B stores sperm temporarily. Okay, let's go to our diagram. So the answer there becomes B, the epididymis. Remember, the function of the epididymis is that it stores sperm temporarily, right? Okay. Number C. Let's go to C. So C secretes testosterone. It secretes testosterone. Already we know the answer. The answer is E, which is testes. So E, testes. So remember, here, read this question carefully because a lot of students, they make mistakes here. So please give the letter and the name. Okay. Now let's go to 1.4.2. Identify part A. So what is part A? Let's go to part A. So part A becomes your prostate gland. Remember, prostate gland is the biggest gland, right? It's the biggest gland when you compare it to other accessory glands. The smallest gland is the corpus gland. Remember, it's the smallest one. So this is the corpus gland, and the leaf-like gland is the seminal vesicle. And the biggest one definitely becomes your prostate gland. It's easy to identify them like that. Becomes easier. Okay. Now let's go to identify the type of gametogenesis that takes place in part E. Identify the type of gametogenesis. So what is gametogenesis? Gametogenesis is the formation of gametes. Genesis means the beginning, right? The manufacturing. So let's go to part E. So part E is our testes. Obviously, we know that the gametogenesis that's there is our spermatogenesis. So the answer there becomes spermatogenesis. Okay, let's go to 1.5. 1.5. The reason I'm moving at such speed is so that I don't waste any more of your time, guys. Right? Okay, let's go to 1.5. The diagram below represents some structures involved in homeostasis in the body. Remember, we said homeostasis is the ability of the body to maintain a constant internal environment regardless of the external changes, right? Or regardless of what's happening externally, as long as the internal environment is constant. Okay, obviously. So how do we analyze the structure? So we can see that our A becomes our pituitary gland, right? A becomes our pituitary gland. Then our D, your D, you have your pancreas, right? At D, you have the pancreas. Then obviously this is part of the skin. So then part C becomes our sweat glands, which are responsible for secreting sweat, right? Then our B is our blood vessels or our blood capillaries. It's either you can say blood vessels or blood capillaries. It's one and the same. Okay, it's one and the same. Let's continue. Let's move to our question. Let's move to our question. Okay. Let's move to our question. But I think we're not done analyzing. Let me just move on. Okay. So here we have our F. Remember, our F is what? It's our adrenal gland. It's just on top of the kidney, right? And E, our E becomes what? Becomes the kidney itself. Okay. Okay, let's go to the questions then. Let's go to the questions. So 1.5.1. It's moving very fast. Okay. Write the letters of the parts that serve as endocrine glands only. Remember, what are endocrine glands? Endocrine glands are ductless. They produce their secretions are secreted directly into the bloodstream, right? They don't have ducts. Does that make sense? And we can see that the pituitary gland is part of A, which is the pituitary gland is part of the endocrine gland, right? A, ish, I'm drawing above now. Okay, A is our pituitary gland. That's our first answer. D is the pancreas. It's not the answer. So the pancreas forms part, right? It's both part of the endocrine gland and the exocrine gland because it has the pancreatic duct and also secretes insulin and glucagon directly into the bloodstream. So it forms part in both glands, right? So it forms part of the endocrine gland, forms part of the exocrine gland. Okay. So our answer is F. It's A and F. So our F is what? Adrenal glands. So our answers respectfully are A and F. but they wanted the names of the glands. So our answer is the pituitary gland, or you can say hypophysis, and the adrenal gland. Okay. Okay, let's go to 1.5.2. Give the letters and names of the parts responsible for the regulation of the body temperature. Regulation of the body temperature. Okay, there's letters and names. Okay, obviously we have our C here, C, which is our sweat gland, right? C, sweat gland, and B, our blood vessels or blood capillaries. Okay, so we're done. We are done. Okay, now let's go to 1.5.3. Name the following with regard to the regulation of water content in the body. A hormone secreted by part A. Let's check our part A. We've already identified part A. Part A is the pituitary gland. And which hormone is secreted by the pituitary gland that deals with the water content in the body? We have the answer is ADH, or antidiuretic hormone. Okay. Target organ E. So target organ E becomes our kidney. So kidney. So target organ E is the kidney. Okay. Let's move. Let's move. Let's move. So let's go to section B. So we're done with section A. Section A is the easiest section where you can collect marks. So don't do a disservice to yourself by not collecting marks in question section A. Right? Let's go to section B. Now section B, these are high order questions where you have to explain yourself articulately, right? Eloquently, and articulate yourself in a manner that is understandable, just to show that you know you can comprehend the theoretical part. Okay, let's go to our structure here. So let's analyze the structure before we go to the question. I love to analyze the structure before we go to the question. So we can see that we have C. Our C becomes what? It becomes our ovum, right? Or you can say in Excel, right? Ovum. Then our, let me see this letter. I can't see this letter. Let me just move. Let me do this. No, it's this like A. So our A becomes our fallopian tube. Fallopian tube because it's one fallopian tube, right? So this is our fallopian tube, the site of fertilization. So this is where fertilization will take place. And you can see that fertilization is about to take place, right? And we have sperms here, right? Swimming towards the egg cell or towards the ovum. And this is our endometrium preparing for pregnancy. So you can see that our endometrium is thicker, vascular, and glandular, right? So the endometrium is thicker, vascular, and glandular. So already preparing for implantation, not pregnancy. Let me not say pregnancy, but implantation. Okay, let's go to our questions. So identify part A. So what is part A? Already identify that it's fallopian tubes. Identify part B. So part B is the ovary. Actually, we did not forgot to identify it. So part B is the ovary, right? Part B is the ovary, and also we can see that this process here, you can see that this process here, where the egg cell or the ovum is released from the ovary, is called the ovulation. Called ovulation. You can see the ovulation process here. Okay. Now let's go to the next question. So B is the ovary. So let's go to 2.1.2. So give two characteristics of the endometrium that make it suitable for implantation. Endometrium, okay? So we can see that our endometrium is glandular, right? It's full of glands, and also it's vascular, it's full of blood vessels. So it's full of blood vessels for nutrition, and also it's thick. You can see the thickness in order to prepare for implantation. So those are the structural adaptations that are visible in this picture, right? So we can see the thick part, right? And also the vascular part. It's filled with blood vessels. So blood vessels are there to serve the purpose of nutrition. Okay, let's go to 2.1.2. B. Give two visible reasons why there is an increased chance of fertilization in this female. Remember, what is fertilization? Fertilization is the fusion of the nucleus of the sperm cell and the nucleus of the egg cell, or the nucleus of the ovum. So let's look at observable reasons or evidence that proves that we are about to, there's an increased chances or an increased chance of fertilization. So we can see here that already the ovum is released. We can see ovulation here. The ovum is released from the ovary into the fallopian tube, and we can see that the sperms are approaching, right? They're approaching towards the ovum. So we can see those two things, right, which increases the chances of fertilization. At this point, I don't think that there can be any doubt that fertilization is about to take place, right? Okay. Now let's move on. Let me just leave it as it is. Move on to the next question. So let's go to 2.1.3. 2.1.3, identify and describe the type of gametogenesis that leads to the formation of structure C. I want you to listen to me clearly. So structure C, let's see structure C, so it's the ovum, right? So the process is oogenesis with the two O's. Remember, it's very important, two O's, right? With the two O's. So this is how I like to start the process, right? So under the influence of FSH, it needs to be under the influence. That's the first point of FSH, right? Or follicle stimulating hormone. And at the onset of puberty, because this process happens after puberty. Before puberty, it does not happen. So it happens after puberty. A diploid cell, right, which is your germinal epithelium cells, the diploid cells, they undergo mitosis to form numerous follicles in the ovary. So they form numerous follicles in the ovary, right? Then one cell inside the follicles, one cell inside one follicle undergoes what? Meiosis. Undergoes meiosis to form four gametes, right? To form four gametes. Remember that it forms four gametes, which three die, right? Three of the four die, and only one matures to become an ovum. And this process happens in a monthly cycle. So it happens in a monthly cycle. Does that make sense? So go back to your oogenesis process, revise it and everything, right? And also recite it as much as I've recited it in this video. Okay. So let's continue. So 2.1.4. In an ectopic pregnancy, the fertilized ovum can become implanted at part A in part A. This normally results in the death of the embryo and may endanger the mother's life. Okay? So explain, the answer is explain why an ectopic pregnancy may result in the death of an embryo. So what is ectopic pregnancy? So ectopic pregnancy is when the embryo, or not the embryo, let me just say the zygote, or I can say the embryo, right, implants at A, right? So when the embryo, or let me just use the embryo, implants at A, as they've already described it, right? So what's the problem when the embryo implants here at A? The problem is that at A is the fallopian tube. A is not structurally adapted for implantation, right? It's not structurally adapted in a sense that it does not contain any blood vessels, so the embryo won't receive any nutrition, right? So definitely the embryo will die there. And also it's not thicker, right, for implantation. It's not thicker for implantation. So the embryo will definitely slip away from those fallopian tubes, and you will result in the death of the embryo, right? And also, it's not glandular, right? Which are the necessary conditions for implantation. Does that make sense? So it's not structurally adapted for implantation in that case, because it's the fallopian tube. It's the site of fertilization. right? So I hope that makes a lot of sense. Hope that makes a lot of sense. Okay, let me just remove this. Okay, let's go down. Okay, so done. Go to 2.2. So 2.2, the table below shows the levels of two hormones during the menstrual cycle rather, of a healthy human female. So we can see here they're comparing, right, or juxtaposing two hormones. They're juxtaposing estrogen level in on different days, estrogen level and progesterone level. Right? So we can see already with the foreknowledge, right, of the menstrual cycle, we know that in the beginning days you have you have your estrogen at its highest, right? In your early days, right? Because the graphene follicle is still developing. So the graphene follicle is still developing and releasing what? Estrogen. But as we move forward, right? As we move forward on your day 16, day 20, you see an increase in the level of progesterone, right? Why do you see an increase in the level of progesterone? Because the corpus luteum is secreting progesterone at this point in time, right? In order to prepare for pregnancy or in order to maintain pregnancy, right? Okay, let's go to 2.2.1. On which day of this menstrual cycle is the level of progesterone the highest? Which day? So obviously we can see that day 20, the level of progesterone is the highest. Yeah, we can see that day 20, the level of progesterone is the highest. And we understand why, what causes this, right? Okay, let's go to the next question. 2.2.2. Name the reproductive hormone that will begin to increase from day 24 of this female's menstrual cycle. So from day 24. So from day 24, as we study this table, let me draw. As we study this table from day 24, we can see that the levels of progesterone are decreasing. So from 15.0 to 5.0, then eventually 0.8. So when the levels of progesterone go down, which other hormone increases? Obviously, we know that it's FSH. So you have your FSH levels increasing because FSH and progesterone, they re- they work with each other antagonistically, right? So they oppose each other. So when the levels of FSH are down, the levels of progesterone are up. When the levels of progesterone are up, the levels of FSH are down, right? So antagonistically, so they oppose each other. Right, so as we can see here in this table that the levels of progesterone are starting to decrease. As they are decreasing, the levels of FSH will start to increase. So the answer here becomes that the levels of name the reproductive hormone that will begin to increase. So the answer here is FSH or follicle stimulating hormone, right? For the reasons that I've just explained. So 2.2.3, use the data from the table to explain your answer to question 2.2.2. So here we can see that there's a decrease in the level of what progesterone, which will stimulate an increase in the levels of FSH. So it means the pituitary gland will then secrete the FSH, right? We secrete more levels of FSH because now it's stimulated by the decrease in the levels of progesterone, right? But let me explain the significance of this negative feedback between FSH and progesterone. The reason why you have your FSH level increasing when the progesterone levels are down or when they're decreasing is so that you can have follicles that are developing in the ovary, right? We can have follicles developing in the ovary because it means that when the levels of progesterone are decreasing, fertilization did not happen. So already the ovaries are preparing for another cycle, for another menstrual cycle. But when the levels, let's say the levels of progesterone remained high at this point in time, from day 24 to day 28, it means that the level of FSH will go down, will remain down or they will stay down. Why? Because it means that fertilization happened. So if fertilization happened, it does not serve any purpose for follicles to develop, to keep developing, because fertilization already happened. So that's the reason. I hope I've explained it to the best of my abilities. Okay. Let's go to the next question. So calculate the percentage increase in the oxygen level from day 8 to day 10. Show your workings. So whenever they say calculate the percentage increase, the numbers that are given in the table are not in percentage, right? Units. So they are in PG slash ML, right? Or NG slash ML. So, but they spoke about oxygen levels. Let me just confirm that they spoke about, okay, oxygen levels, right? So this is the formula that you're going to use for a percentage increase. So it's the new value, not the new value minus the old value divided by the old value, then multiply by 100, right? Okay, let me just do that again. Let me just write it down. I wish I could write it here. Let me try to write it here. So what's our new value? So our new value, let's look at the days. Let's confirm the days first. So I'll do it on the side so that you can see it. Compare this, confirm the days first. So from day 8 to day 10. So day 10, we can see in day 10 or day 8, it was 70, right? It then increased to 280, right? On day 10, right? You can see there. So now this is how we're going to do it. What's our new value? New value is 280. Trying to complicate it to write with this pen. So 280, right? I hope you can see that. Minus 70, minus 70, minus 70, right? Divided by 70, and multiply by 100. Or multiply by 100 divided by 1, but it's still the same thing. Multiply by 100. You know, when you're converting something to a percentage, you need to multiply by 100. Right? Multiply by 100. Okay. So what's 280 minus 70, which is your answer is 210, right? Right, so what's 210? 210 divided by 70, which is 3. The answer is 3. Right, so let's say, so here it's the answer. 280 minus 70 is 210. 210, right? 210. Then 210 divided by 70. 210 divided by 70. Multiply by 100. Just by 100. And this thing's writing very slow. So 210 divided by 70, I think it's 3. 3. 3. Then 3 times 100. 3 times 100. 3 times 100 is 300. So the answer is 300%. Right. The answer is 300%. And so just make sure that you carry a calculator, right? Whenever you writing last answer. And also a protractor in case they ask you to draw a pie chart. So remember, so it's the new value minus the old value divided by the old value multiplied by 100. So that's the formula for percentage increase. Right, that's the formula. Okay, now let's go to 2.2.5. How would the progesterone level differ after day 20 if this female was pregnant? Obviously, we know that the levels of progesterone will remain high, right? So in day 24, you can see here. So if this female was pregnant, then the levels of progesterone will be high, right? Here, with day 24 and day 28. So they will remain high, right? So we know why they remain high. The reason why they remain high is because the corpus luteum continues to secrete, right, more progesterone. It does not disintegrate or degenerate. Okay, let's go to 2.2.6. Explain what causes the change in the progesterone level stated in question 2.2.5, right? So it's because the corpus luteum continues to increase in size or continues to be constant in size and continues to secrete more progesterone. So that's the answer. So nothing complex there. Okay, let's move on to the next question. Okay, I will just read the statement very quickly, right? 2.3. Cataracts. Already we know what a cataract, right? Cataracts is when your lens becomes clouded, right? It becomes clouded. So it's when it becomes clouded. And when it becomes clouded, it means that it cannot allow light to pass through the lens, right? Because remember, the lens is transparent, right? Why is it transparent? So that it allows light to pass through, right? After that, the image is formed on your retina, right? The light falls on your retina, specifically the yellow spot where a clear image is formed. So cataracts, right, prevents or gets in the way of that function to be fulfilled, right, or to be performed. Okay, so let's read the statement. Cataracts are primarily formed when protein structures in the lens of the eye start to disintegrate and clump together. Cataracts is the most common cause of blindness and the main cause of vision loss in people over 40 years of age. Two types of cataracts that occur are nuclear cataracts and subcapsular cataracts. In nuclear cataracts, the cataracts form in the central region of the lens and gradually spread outward. In subcapsular rather, cataracts, the cataracts develop at the back of the lens and it is most common in people with diabetes. Okay, so according to the 2.3.1, let's go to 2.3.1. According to the passage, which type of cataract is associated with the disorder of the pancreas? Obviously, we know that the disorder of the pancreas is diabetes mellitus, right? And we know that they've told us in the extract that subcapsular cataracts, right, they develop in people with diabetes. Okay, let's go to 2.3.2. Use information in the passage to explain why cataracts cause vision loss. So why do they cause vision loss? So we know that they explain to us in the extract that it's formed, right, by protein structures in the lens of the eye starting to disintegrate and clumping together. And that disintegration and that clumping together causes a cloud in the eye, or it causes a cloud in the lens, right? And when it causes a cloud in the lens, it creates it, how can I put this? It prevents the lens from being transparent, right? So, and when it prevents the lens from being transparent, it means that the lens cannot no longer allow light to pass through because now it's no longer transparent, clouded by this clump, right? So it's clouded. So when it's clouded, it cannot allow light to pass through. And when it does not allow light to pass through, it means that light won't get through to the retina where a clear image is formed. And when a clear image is not formed, then it means that the stimulus or the light stimulus is not converted to an impulse. And if it's not converted to an impulse, it's also not taken by the optic nerve to the cerebrum. So there's no interpretation, right? So you need to build up your answer whenever you're answering a question, especially when they say 5 marks. So build up the answer. right? You can even start by explaining what cataracts are. And after explaining what cataracts are, you explain how do they prevent the ability of the lens to perform its function. And after explaining that, you explain the consequences of cataracts as I've explained it, right? So that's how you approach a question, especially longer version questions like these. Let's go to 2.4. So 2.4.1. Okay, 2.4 states that a man is long-sighted. So we know what long-sightedness is, right? Or hyperopia. So hyperopia or long-sightedness is when you see things that are far, right? But you can't see that things that are close to you, right? Or things that are less than 6 meters. But you can see those that are 6 meters away from, or more than 6 meters rather. So explain how 2.4.1 the structure of the eyeball affects his vision when he reads a book without glasses. So the eyeball, we know that the eyeball in people who are long-sighted is shorter than normal. So the eyeball is shorter, and that causes the light to fall behind the retina, right? And when the light falls behind the retina rather, it causes a blurry picture. So that the book he's reading will be blurry because the eyeball is shorter than normal, which causes the light to fall behind the retina. And when the light falls behind the retina, because remember, the light is supposed to fall in on the retina, not in front, not behind, but on the retina, right? But when it falls behind, right? As in the case of long-sighted individual, when it falls behind, it creates a blurry vision. Okay, so let's go to 2.4.2. Wearing glasses with convex lenses will improve his vision when he reads a book. So wearing glass with convex, remember convex lenses. So convex, it means that it's curving outwards. So it means it's curving outwards. Does that make sense? And anything that curves outwards, like the cornea or the lens, it means that it will refract or bend. Refraction is bending. It will bend the light, right, inwards. That's focusing the light on the retina. So it will bend the light more inwards, right, focusing the light on the retina because that's where the image is supposed to fall on. It's supposed to fall on the retina. Okay, let's go to 2.5. Didn't I jump any question? No, I didn't. Okay, let's go to 2.5. So 2.5, the diagram below represents a neuron with a myelin sheath, right? And a neuron without a myelin sheath, right? Already we know, we can analyze this question that the neuron that does not have a myelin sheath is the neuron of a person who has uh, multiple sclerosis. But the one with the myelin sheath, that person does not have, right? Multiple sclerosis. Remember, what is multiple sclerosis? Multiple sclerosis is when your immune system attacks the myelin sheath up to the point that it disintegrates or it degenerates. And what's the function of the myelin sheath? It accelerates the transmission of impulses. So it means that when a neuron does not have a myelin sheath, the transmission of impulses becomes slower. Okay, now let's just identify the structures before we go to the question. So you have your A. So what is our A? Our A is our axon, right? Because if we don't have a myelin sheath, we're left with the axon. So our B is our terminal branches of axon. It's just an extension of the axon. So our C becomes our dendrite. And what type of neuron is this? Because we see that the neuron is multipolar, right? It's multipolar, it has multiple poles, right? These are poles, they're called poles. You can see that it's multipolar, right? You can see that it's multipolar, which proves that this neuron is a motor neuron. It's not a sensory neuron. Sensory neuron is unipolar, right? There's only one pole. But when it comes to a motor neuron, it's multipolar. Okay. Okay, let's go to the question. Identify the type of neuron shown in the diagram. Definitely the type of neuron shown in the diagram is the motor neuron. Okay, let's go to 2.5.2. Give one visible reason for your answer. I've already given you multiple reasons, right? Not multiple, but one reason. So the reason is that the neuron is multipolar. And another reason you can refer to the position of the cell body is at the end. The position of the cell body is at the end of the neuron, right? So it's at the end of the neuron. Okay. Let's go to 2.5.3. Describe the function of the type of neuron identified in question 2.5.1. So what's the function of the motor neuron? So the function of the motor neuron is that it collects or it transmits impulses from the CNS, right? CNS can be the spinal cord or the brain, from the CNS to the effectors. The effectors are your muscles, right? So that's the function. So it transmits impulses from the CNS or the central nervous system to the effectors, which are your muscles. Okay. So use the letters A, B, and C to indicate the direction in which an impulse moves through the neuron. In which an impulse moves through the neuron. Okay. So the first thing that you do here, you identify the dendrites first. So you have your dendrites here. So these are dendrites. So that's where the impulse starts. Then after that, you identify your axon, because the axon takes impulses away from the cell body. So the dendrites, they channel or direct impulses towards the cell body. Then the axons, they take the impulse away from the cell body. Then you have your B. So this is the direction of the impulse. Right, this is the direction of the impulse. So it's going that side. So it's from CAP. So that's the direction of the impulse. But firstly, you need to identify your dendrites. Then it becomes easy for you to identify the direction of the impulse. So direction, so your answer it becomes what? CAP. So your answer becomes CAP. Okay, 2.5.5. Name the disorder associated with disintegration or degeneration of the myelin sheaths of neuron. Okay, multiple sclerosis. Already mentioned the disorder above, right? Okay, now let's go to 2.6. 2.6. In the human body, the axons of the myelinated neurons have much smaller diameters than the axons of the unmyelinated neurons. Right? We know myelinated neurons, they have a myelin sheath. Unmyelinated neurons, they don't have a myelin sheath. Right? So the graph below, right, show the speed of nerve impulses in myelinated neurons and in unmyelinated neurons with different axon diameters. So already we know that the ones with myelinated neurons, the speed will be faster as compared to the ones that are unmyelinated. That's just the common sense of it, without even looking at the tables there. So we can see the axon diameter, right? The axon diameter of the unmyelinated neurons, as they've already mentioned above, is more than the one of the myelinated neurons. But look at the speed, right? So the speed of the myelinated neuron is more than the speed of so this one has a high speed myelinated neuron, but this one, unmyelinated neuron, has a lower speed. We understand that because it does not have a myelin sheath, right? I'll mention the function of the myelin sheath again. So the function of the myelin sheath is to accelerate the transmission or speed up the transmission of impulses. So 2.6.1, give the axon diameter in right, at which an impulse moves at 20 m per second, minutes per second, meters per second, m per second, in, right? I'm just butchering those units, man. Okay, in 20 m per s, right? Let me just say that it makes it easy. Okay, okay, let's go at 20. So we need the axon diameter at 20. For a myelinated neuron. So let's look at 20. So there is 20 here. So we need the diameter. So there is 20, right? 20. So this is 5. So the diameter, it's 5. So it's 5 UM, right? It's 5. Then for A, let's go to the next question. Let's go to B. So the answer for A is 5 UM. So B, unmyelinated neuron. Let's look at 20 ms for the unmyelinated one. So 20, there's 20 here, 20 here. Right now, let's see the diameter of this one. Okay, see, diameter is like 800 UM. So this one, so it's 800. Wow, I'm impressed. I just drew a straight line without using a ruler. My hand is more stable, right? It's the most stable uh, hand in the world. The most stable indeed. It's the most stable. Straight. English is running out now. The most stable. Stable. The most stable hand. Right, so it's the most stable hand. So that's the right uh, language. Right. Okay, so we're done with 2.6.1. So 2.6.2, let's go to 2.6.2. So 2.6.2, use the graphs to describe the following. Difference in impulse speed for unmyelinated and myelinated neurons. Okay, so difference in speed. We can see that the myelinated neurons are faster than the unmyelinated neurons, or the unmyelinated neuron is slower, right, as compared to the myelinated neuron, either or, right? This word is a tongue twister, eh? So let's go to B, the relationship between axon diameter and impulsing speed, right? Axon diameter and impulsing speed. So let's look at this one, right? So the diameter, right? The lower the diameter, right? The lower the diameter, because we can see that the diameter of the myelinated neuron is small, right? The diameter of the myelinated neuron is small. Hence, whereas the speed is faster, right? For the myelinated, it's faster. The speed is faster, right? So the diameter is small, and the speed is faster, right? But when it comes to the unmyelinated, you can see that the diameter is very large, right? But the speed is very slow, right? Here, the diameter is small, but the speed is fast, right? Here, the diameter is large, but the speed is slow. So we can see that the relationship between the axon diameter and the speed is inversely proportional, right? Or indirectly proportional. So when the diameter, so the smaller the diameter, the faster the speed. Or the bigger the diameter, the slower the neuron becomes, or the speed. Right, so that's the relationship I'm seeing here. That's the relationship I'm seeing here when I analyze both graphs. Right. Okay, so B, we're done with that question. Now let's go to question 3. So question 3, 3.1. The passage below describes the breeding habits of vultures. So vultures, okay? Vultures are birds of prey that feed on flesh of dead animals. A female vulture lays only 1 to 2 fertilized eggs, which are then protected by both parents during incubation. Incubation is the period from the time an egg is laid up to the time it hatches. So vultures have a short incubation period of 32 to 45 days. This short incubation period is typical of birds displaying altricial development. After hatching, the parents feed the young chicks as often as 20 times a day. Okay, so altricial development, we see that altricial development, they can walk, right? It's a development where the young cannot walk, they cannot see, they cannot eat. So really they've told us it is artificial development. Let's go to the questions. Give one reason from the passage why vultures are considered as oviparous. Remember, ovipary, right, is where the eggs are laid, and we see that in the extract they told us that the eggs, right, the vultures they lay eggs, right, until so they laid the eggs are laid, right, until they hatch. So they lay eggs. So that's the reason why they're considered oviparous, because ovipary is a reproductive strategy where birds lay eggs. Eggs are laid, right? Okay, B. Let's go to B. Give one reason from the passage why vultures have a high survival rate even though few eggs are laid. The reason why is because the eggs are protected by both parents. So that's the reason why. During incubation. So the eggs are protected by both parents in order to prevent predators. Okay, so this was an easy paper. This was an easy paper. So let's go to 3.1.2. Fertilized Fertilization in vultures takes place internally. State two advantages of fertilization, internal fertilization. So internal fertilization is fertilization that happens inside the female's body. So what are the two advantages? Is that the eggs are laid inside, right? So they're laid inside. So it means that they're protected from predators because the eggs, or fertilization rather, not laying of eggs. Let me just correct my language here. So fertilization happens inside the female's body. So the sperm cell and the egg cell, or the ovum, are protected. The gametes are protected internally. Does that make sense? And also the gametes, they don't have high chances of drying out. Right? They're in a moist environment as compared to when the gametes are laid outside of the mother's body. So they stand a chance of dehydrating or drying out. Right? They stand a chance of dehydrating or being eaten by predators. But internally, there's moisture internally. So they will be moisturized through and through until fertilization happens. Right? So those are two advantages of internal fertilization. 3.1.3. State two characteristics of chicks that display artificial development. Okay, artificial development, their eyes are closed, right, and they cannot feed themselves. They cannot walk by themselves. They are dependent on the mother, right? Even though I mentioned more, but I was just giving you more options, right? Let's go to 3.1.4. Explain why birds with artificial development would have short incubation periods. So why birds with artificial development will have short um, incubation periods? Let's go to the extract. Incubation, during incubation. Okay, so the reason why birds with artificial development would have short incubation period is because they don't feed the young, right? Whenever they're still incubating them, but they feed them after hatching, right? They feed them after hatching. As they mentioned in the extract, that they feed them 20 times a day, right? So the reason why it doesn't last long is because they feed their young after hatching. So they don't spend a lot of energy in feeding the young during incubation, but they spend most of the energy after incubation. Hence, most of the young, they are born without being developed, right? Without without being able to walk by themselves, without seeing, right? So that's the explanation. That's the sensible or logical explanation I can give to this question, right? Okay. Let's go to 3.2. So 3.2, diagrams below represent the pupillary mechanism in the human eye. So really it's easy to analyze the structure. So what's structure Y? So what is structure Y? So structure Y becomes your iris, right? So structure Y is the iris, right? And structure X becomes the pupil, right? So structure X is the pupil, okay? So we can see that the person is moving from bright light, because you look at the size of the pupil. When the pupil is smaller in size or it's constricted. So it means that the person is exposed to bright light. But when the pupil is dilated or wider, means now the person is exposed to dim light. And what happens when a person is exposed to bright light, right? What happens to the muscles? Remember, in the iris, we have two muscles. We have the sacral muscles and the radial muscles. So what happens to the sacral muscles when the person is exposed to bright light? So if the sacral muscles contract, already you know when the sacral muscles contract, the radial muscles will relax. And when the radial muscles relax, the pupil will constrict, right? Allowing less light to enter the eye. So what happens then when the person is exposed to dim light? So the sacral muscles will relax and the radial muscles will contract. And when the radial muscles contract, it causes the pupil to dilate, right? Or become more wider, allowing more light to enter the eye. Okay, let's go to 3.2.1. Identify part X. So part X becomes your pupil. B, identify part Y. Part Y becomes the iris, not the muscle, right? They did not show us any muscles here. So the closest answer to this is what is the iris Y. Explain why the pupillary mechanism is considered to be the reflex action. Because it's an automatic rapid response to the light stimulus. Right? It's responding to the light stimulus and it's automatic and rapid. Right? It's automatic and rapid and it's responding to what? To a stimulus. So it's an involuntary action. So that's the explanation that you need to give here. Right? That's the only reason why it can be considered as a reflex action. Remember, what is a reflex action? Reflex action is an automatic, right, rapid response to a stimulus. In this case, the iris, right, or the pupillary mechanism, or the iris, let me just say the iris, is responding to the stimulus, to the light stimulus, and it's rapid and it's automatic. It happens without you even thinking. Okay. 3.2.3. Name the two effector muscles that are involved in the pupillary mechanism. As we've mentioned before, have your sacral muscles and the radial muscles. Please don't confuse sacral muscles and radial muscles. Right? Please don't do that. 3.2.4. Explain the significance of the change in the diameter of part X and of part X from diagram A to diagram B. Right? So as we've stated before, the importance, so the significance is the important, right? So as we've stated with the sacral muscles, they relax, and the radial muscles in diagram B, they contract. Why do they contract? So that the pupil becomes wider. Why does the pupil becomes wider? So that more light enters your eye. Why do you need more light to enter your eye? Because you're exposed to a dim environment, right? So that you can see clearly. A clear picture can be formed on the retina because you are in a dim environment. So that's why it's important for the pupil to become wider or to become dilated so that more light is allowed to enter the eye. So in this case, you need to understand your pupillary mechanism, both in bright light and dim light. Okay, let's go to the investigation. An investigation was conducted to determine the effect of insulin on blood glucose levels. The procedure was as follows. The two groups of 10 people each were selected. Two groups of 10 people each, that's the sample size, but it's the smaller sample size, right? They could compromise the reliability of the investigation. It's just a small sample size, 10 people, two groups of 10 people each. Okay, one of the groups consisted of healthy individuals, and the second group, which served as a control, consisted of people with diabetes, right? So already they told you the control, right? Which are the people who have diabetes, right? So let's read the statement. I want to read the statement again. An investigation was conducted to determine the effect of insulin on blood glucose level. So what's the independent variable there? The new independent variable is insulin, right? And the dependent variable are your glucose levels. So the statement will tell you the independent variable and the dependent variable, right? Remember, the independent variable is constant, does not change, right? And the dependent variable changes, right? Okay. Each group was given 75 grams of glucose to ingest. So now we are improving or ensuring validity. We need to give each group the same amount of glucose, right? Because if we give them different glucose levels, right? Now it will compromise the validity of the investigation. Let's go to the next point. Their blood was tested again after 60 and 90 minutes to determine the levels of glucose and insulin. Okay. Note the normal level of glucose in the blood is between 3.9 moles per liter and 5.6 mole per liter. Okay. The normal level of insulin in the blood 60 minutes after glucose ingestion is between 18 UM per liter and 276 UM per liter. Okay, then let's go to the question 3.3.1. State the purpose of the control in the investigation. So the purpose of the control is to compare the results, right, between the group that's part of the experiment, right, between the group that's part of the experiment and the control itself, right, is to compare the results or is to look at the changes in the glucose levels or the effect of the level insulin on the blood glucose levels. So the purpose of the control is to compare. So never forget that. It's to compare, right? Describe two ways in which insulin decreases blood glucose levels. Okay, so let's look at how insulin decreases blood glucose level in general. So this will go back to the knowledge of your endocrine system, right? Or homeostasis or negative feedback, right? So blood glucose level. So when your blood glucose level is high, right, what will happen? Your pancreas will secrete insulin, and the insulin, right, will go to your liver. Remember, the target organ is the liver, right? And in the liver, it will stimulate the conversion of glycogen or of glucose to glycogen, right? Glucose to glycogen, and please don't confuse glucagon and glycogen. Right? Glycogen is a stored carbohydrate, so it will stimulate the conversion of glycogen to of glucose to glycogen. Rather, I don't know why I keep on making this mistake. Apologies for that. The conversion of glucose to glycogen. Then your levels of glucose will come down. Right? Does that make sense? I hope that makes a lot of sense. Okay, let's go to the second question. Which group, X or Y, consists of healthy individuals? Let's see. Obviously, we know that the individuals that are not healthy, their glucose levels will be above normal. Right? Hence, they told us the normal levels of glucose in the blood. So the normal levels of glucose levels in the blood are between 3.9 and 5.6. So let's look at the abnormal glucose levels in the table there. See, time after glucose. So we see here time after glucose ingestion. So group X 16. Okay. Okay. Average level of glucose in the blood. Okay, average level of glucose in the blood. See 3.9. So we're looking for levels that are between 3.9 to 5.6. So after ingestion, okay, yeah. See between okay minutes. So after 60 minutes, okay. So this is just way above normal. Just way above normal for group X. Can you see which is way above normal? Because the normal The normal glucose levels in the blood is between 3.9 to 5.6. For group X, this is way above normal because after 90 minutes, after 60 minutes, it was like 8.9. After 90 minutes, it was like 8.7, right? So it's way above. So the normal or the healthy group of individuals is group Y because it's closer, it's between 3.9 and 5.6, even though it's between there, right? You can see that the insulin here, so there was no insulin here. You can see that in group X, the insulin did not affect the level of glucose in the blood, whereas for group Y, the insulin affected affected the levels of glucose in the blood in a big way, right? Okay. And my battery is about to my laptop is about to shut down. Use the data. Okay, I already explained the answer for 3.0.3. I don't know whether I can finish this question paper or should I release the remaining questions. I'll release the remaining questions in the next video, guys, because my battery is about to shut down. About to shut down. My laptop actually is about to shut down. So I'll release the next video, right, so that we can analyze the question together. Thank you.